Hey Bio30s, gotta stay in shape here, get the workout in whenever you can. But while I have you, let's finish up chapter 20, talk a little bit about biotechnology and DNA mutations. So, when we talk about biotechnology, there's a few things we need to discuss here. So, there's a lot of cool stuff and a lot of cool research going into DNA technology, DNA enzymes, things like that. But we won't be able to go through all of it. I'm just going to go through kind of the main ones, not the most important ones, but the ones that I think are pretty cool and unique. So, first one is recombinant DNA. So, recombinant DNA requires you to essentially take DNA from one species and then insert it into another species. And yes, I know that sounds very science fiction-y, and it sounds like Jurassic Park, where they took dinosaur DNA and frog DNA and melded it all together. But it can be done in a way that is super useful for humans. So let me explain how it's done, okay? So, you're gonna take a restriction enzyme. I like to think of the restriction enzyme as essentially molecular scissors, okay? So you're going to do a little bit of molecular arts and crafts. You're going to take the restriction enzyme and you're going to cut out the section of DNA that you want to copy. So in this case, we're going to go into a human genome and we're going to cut out the section of DNA that makes the hormone insulin. So this would be from an islet cell in your pancreas. So you'd take an islet cell, you'd analyze that islet cell, and you'd cut out the segment of DNA that makes insulin. You would then go to a bacteria, let's say E. coli. And remember, not all E. coli is bad or harmful. You'll go to that E. coli and you'll notice this little ring here. That's called a bacterial plasmid. Bacteria are very special in a sense that they have a ring of DNA called a plasmid, and it's separate from their genomic DNA. So it's separate from their big mass of actual, they don't have a nucleus, but it'd be a big mass of DNA right there. I guess something. So then anyway, you would take that plasmid, use that same enzyme to cut out the same section on that plasmid. You would then use DNA ligase, which is like molecular glue, to glue that human DNA into the bacterial DNA. And the reason this works is because DNA is universal. A's and T's and G's and C's are found in all species. It's just, once again, how much, how many sequences, things like that. So now what you've done is essentially inserted the human gene for insulin into a bacteria. Why would you do this? Because every time this bacteria replicates, that's probably going to be every 30 minutes, every couple of hours, you get insulin. So now you're essentially using bacteria to make insulin for you. So that's one of the cool technologies out there related to DNA. It's called recombinant DNA technology. Another DNA technology out there that has recently been very, very important in criminal investigations, as well as testing for usually paternity, but you can sometimes test maternity as well, testing who the mother is DNA profiling. So what you're going to do is, if you have DNA at a crime scene, so if you have DNA at a crime scene, you can actually test that DNA or compare that DNA to your suspects. And if they match up, you know that suspect was at the crime scene. Or subsequently, you can test that DNA against, say, a few guys' DNA to figure out which one was the father. Or if you're trying to figure out both parents, you would test it against the paternal and the maternal DNA to figure out which set of parents that child would belong to. So how it's done is pretty interesting. You'll take a DNA sample, you'll amplify it with PCR, which we'll talk about in a second. Then you'll use those same restriction enzymes to actually cut it into various lengths and sizes. Then you put all that DNA into something called gelphoresis. So you'll put it into electrogelphoresis. And then you'll load the DNA into these wells, the gel is kind of like Jello, and you'll turn on a power current. And what happens is DNA is negatively charged because of the phosphate ions. So phosphate makes DNA negatively charged. So it will move from the negative side to the positive side of the gel. So the DNA will migrate through the gel. And the bigger pieces will stay closer to the top of the gel. The smaller pieces will go to the bottom of the gel. And now what you can do is you can analyze them. So let's say this is one segment of, let's say this is one section of DNA here, right? So we have a band here and a band here. Let's say we loaded another one into this well here, and we got a band there, and we got a band there. Notice how these bands, they match up. So now those bands match up with one another, so we can tell that that DNA matches. And once again, you can use that at the crime scene for testing for if you're related to somebody or not. So a lot of different applications for this technology. I mentioned I tell you what PCR does. PCR stands for polymerase chain reaction. Usually when you find DNA at a crime scene or when you want to do a DNA test, you don't need a whole lot of it because you're going to run it through a few rounds of PCR, which is essentially a DNA replication amplifier. So you take your little bit of DNA, you run it through a few rounds of PCR, and then essentially you're going to have millions of copies of that DNA. And the cool thing is, you're just copying DNA. So it's just like running paper through a printer. You're just copying DNA, and now you have lots of DNA to do multiple tests with it and do multiple examinations of that DNA. Now, how PCR works is pretty cool. You're going to take that DNA and you're going to add primers, enzymes, anything you normally would use for DNA replication, but you're also going to add Taq polymerase instead of DNA polymerase, and here's why. To break apart those hydrogen bonds, you need to heat the DNA to 94 degrees Celsius and then cool it to 72 degrees Celsius. Our own enzymes, they don't function at 72 degrees Celsius. However, there's a bacteria that live in hot springs, that's where we get the Taq polymerase from, that are able to replicate their DNA at very, very high temperatures. So we use the Taq polymerase to do the copying because of that high temperature. And then we essentially just copy to copy, copy, copy. We get millions of copies of that DNA, and we can use it to do different tests, run some gels, things like that. Gene therapy. Gene therapy is super cool. So gene therapy is essentially editing genes to fix problems within DNA. So let's say you have a disorder because you have a defective gene, like something's wrong with your DNA, you have a mutation that's causing you some issues. Here's what you can do. You can use a DNA vector. We actually use viruses for this, funny enough. So you use a virus, we insert the good DNA into the virus, okay? Now the virus comes along and will attach itself to that cell. When it attaches itself to that cell, what's gonna happen is the gene is going to be injected into that cell with the insufficient or the damaged gene. And what's going to happen is the damaged part or the ineffective part is going to be cut out, and they're going to replace it with the good gene. So it's essentially cut out the bad, replace it with the good, and now this cell should work again properly. And guess what? Every time this cell replicates, it now has that fixed gene. Every time this virus replicates and transfers to a new cell, it will fix that cell. So now your body's essentially fixing itself from the inside out. This is used a lot of times with genetic disorders that can be solved through gene therapy. Cancer treatments, gene therapy has shown some pretty promising results. It's not always a guarantee, of course, but there's been some really promising results using essentially this gene therapy to edit one's DNA and fix some problems. But this leads into kind of an ethical debate here, right? So far, we've just been editing somatic cells, so cells that have already been formed or are already present in a person. What about if we were to edit the germline? What do I mean by germline? Well, remember those three germ cells, the ectoderm, mesoderm, and endoderm? What if we edit a cell before it's actually an embryo or a fetus or a person that has been born? So what if we edit somebody while they're still in the very early stages of developing? This has caused a lot of controversy because this could lead to the issue of designer babies, where you're essentially designing what your baby will look like. You go into their genes and you edit, I want it to have blonde hair, or I want it to have green eyes, or I want my baby to be this tall. And you edit their genes before they're born, giving them the desired traits that you're looking for. And of course, there's a lot of ethical issues related to that. Right now, it's banned in Canada, banned in most countries, but that doesn't mean it hasn't happened. I think a few years ago in China, 2 babies were actually edited to be resistant to HIV, and they were born with that resistant gene, allowing them to be resistant to HIV. And then they were part of a clinical or part of a study where dads who had HIV, and then they used these babies as part of the test. So while they were embryos, their dad had the HIV virus, and then they edited the embryos to make sure that these kids wouldn't get the HIV virus while they grew and developed. So it was an interesting study, but highly, highly unethical, right? At this point, editing somebody's germline or editing them before they were born is against the law, completely unethical, but it does raise an interesting debate as to, well, if it were legal, or if this were able to be done, would you do it? It's just kind of a question out there that you can think of for your own. All right, I want to end on with some mutations, and then we'll be all done here. So, mutations. What happens when we get a mutation? A mutation occurs in your DNA when a mistake happens, right? Now, sometimes these mistakes can be good. So sometimes we can actually have a beneficial mutation, right? But that's pretty rare. It's pretty rare to make a mistake and have it work out pretty good. Most of the times you make a mistake and it's like, oh man, I made a mistake, and things go from bad to worse. So let me paint you this picture, okay? Let's say you were supposed to have TTT, but it mutates into TTG. Now, one change. What could happen? Well, let's take a look. Your mRNA should have been AAA, but instead you get AAC. Your amino acid should have been lysine, but instead you get asparagine. So wrong mRNA, wrong amino acid, wrong polypeptide, wrong protein. Before you know it, you have yourself a disorder because of it. And an example of this would be sickle cell anemia. Sickle cell anemia is just one change in your DNA sequence that gives you the wrong amino acid. All the other amino acids are correct, but that one incorrect amino acid leads you to have something like sickle cell anemia. So, most of the time mutations are bad, but there are the odd cases where mutations can actually be beneficial. Let's go through some types of mutations. So, we have point mutations, which is changes in a single base pair of a DNA sequence. They may or may not change the sequence of amino acids, because we can have silent mutations where even though we had a mutation, the protein is made anyway, no changes, no problems. We can have a missense mutation, like I talked about with sickle cell anemia, where only one amino acid is changed. We can then have a nonsense mutation, where we change one amino acid into a stop codon. So this is usually lethal to a cell because we don't build the full protein. We stop abruptly midway through the protein synthesis process. Then we can have mutations that affect an entire gene. So we can delete one or more nucleotides, and this completely changes the entire protein. We can also insert one or more nucleotides, and this can change the entire protein as well. They're called frame shift mutations because from the point of the insertion or the deletion, everything after is different. So it's a completely different protein. So let me show you some visuals of what these look like, okay? I'll do this. So here's your regular strand. Here's a silent mutation, okay? So we changed this amino acid here, we changed this nucleotide there, but we still got the same amino acid. So our amino acid didn't change. That's a silent mutation. And this is why it's important that the genetic code is redundant. So even if we have these mutations, they don't result in any changes or differences to the protein. Missense mutations, we make a change here, and now we've changed one amino acid. The rest of the protein is the same, minus this one amino acid change. Nonsense mutation, we make a change, and now it's changed into a stop codon. So now we've changed that into a stop codon, which is a problem because our protein was never formed, which usually is quite bad, quite lethal for the cell. You'll notice a deletion mutation can be a singular codon or a singular nucleotide, or an entire codon right here. So this could be the entire codon, or just a single nucleotide you delete. And an insertion is inserting a codon or a nucleotide into a spot along the DNA. And like I said, that's a frame shift mutation. Frame shift mutations meaning that from the point of the deletion, so right there, or the insertion, everything after is different. So now the entire protein is different. So that's it for chapter 20. As we move along, we're going to now move into the genetics portion of Bio30. So we're going to talk about how we use this DNA. We know how it makes proteins, but now we're going to talk about how we inherit our traits and how things are passed on from person or from parents to offspring and the probabilities behind all that. So thanks again for watching, and we'll chat soon.